Everyone, welcome back to the MOOCs course on Advanced Analytical Technique. We are now discussing the second unit of this course. The title is Molecular Luminescence Spectroscopy. This is the second lecture of the 6th week. So today I will discuss about the methods of obtaining a fluorescence spectrum. There are three methods that are used to study the fluorescence spectrum of a particular sample, or I should say three methods for the fluorescence measurement of the given sample. The first is called as the direct method, then we have an indirect method or the quenching method. In case of direct methods, they are applied to fluorescent molecules only. Means the molecules that are naturally fluorescent can be analyzed by this particular method, because as the name is reflecting, it is a direct method. So the compounds which are naturally fluorescent, like fluorescein, like luminol, like we have rhodamine, coumarin, these compounds are naturally fluorescent, so we can identify these compounds only, and therefore direct methods are the most sensitive methods. They are very specific methods that can be applied to a very small fraction of molecules, because majority of the molecules do not fluoresce, and therefore two other methods have been developed, called as the indirect method, also known as derivatization method, and then we have quenching methods. Indirect method of fluorescence measurements, also known as the derivatization method. This method is used for compounds that do not fluoresce naturally, and that this is the beauty of this technique, that even those compounds which do not fluoresce, those compounds can be analyzed by this technique. And in this method, what we do is we add fluorescence tags to those compounds which do not fluoresce, making them artificially fluorescent compounds. So as it is written here, this method is used for compounds that do not fluoresce. So non-fluorescent compounds, they are converted into their fluorescent derivatives. By using specific reactions, we carry out some reactions in which some fluorescent tags, some specific groups are incorporated into these compounds, so that they becomes fluorescent compound. For example, isocyanate groups, SCN group, are frequently used to create fluorescent tags in molecules, because many aromatic compounds are fluorescent, so we add aromatic rings into the compounds, making them fluorescent compounds, making them artificially fluorescent. Clear? Alternatively, if we are not incorporating these groups into a non-fluorescent compound to make it fluorescent artificially, what we can do is we can add some fluorescent dyes. These dyes attached to specific sites. Sometimes we use those dyes which goes into the sample solution and associate itself with the analyte, or it binds to the specific site of an analyte. For example, if we want to analyze a protein, so we add those dyes which goes and add to that particular protein. However, if in case we do not want to analyze that protein, we already know that a protein is present, we want to analyze a particular site of that protein which is infected. So what we can do is we identify the groups that are present on that particular site, then we select that dye which can go and react with those groups and finally attach to that particular specific site, so that when UV light is illuminated on the sample, that particular site appears to glow by the phenomena, and that particular site appears to glow by the phenomena of fluorescence. Clear? Now these fluorescent dyes include fluorescence, fluorescein is used, we also use rhodamine, cyanine can also be used. Even some other fluorescent agent like 4-nitrobenzofuran, orthothialdehyde, and anthracene carboxylic acid. And if you remember, I told you that many of the aromatic compounds are fluorescent, and if you look at these examples, nitrobenzofuran aromatic, orthothialdehyde again aromatic, anthracene carboxylic acid again aromatic. So these are the structure of these compounds: anthracene, carboxylic acid. This is anthracene carboxylic acid, which is a fluorescent compound and an aromatic, which is a fluorescent compound and aromatic in nature. Aldehyde, and then we have nitrobenzofuran. So these compounds are fluorescent compounds that are basically used to convert or derivatize a non-fluorescent compounds into a fluorescent derivative. So what we do in this derivatization process, the analyte is extracted. First we extract the analyte from the sample matrix, and then the analyte is allowed to react with the derivatizing agent under optimum conditions of pH and temperature, so that the particular reaction occurs to its optimum level and we get the results what we were expecting. The molecule should go and react with the analyte directly, or it should go and bind to a specific site of that analyte. The unreacted derivatizing agent is then separated also by the chromatography, because if that derivatizing agent is still present in the sample, it will also, because it's a fluorescent compound, it will also give its own spectrum that will complicate the actual spectrum. So what we do is, for example, if we have an analyte over here and it has a specific site, and we want our analyte, we want that the fluorescent derivative should attach to this site, we add some fluorescent derivative to it. It goes and react to this particular site, forming a structure, forming a fluorescent tag. Now if some derivatizing agent is still present, what do? We remove it by chromatography technique. And you will learn about the chromatography in the next few lectures. In the third and fourth unit of this advanced course on analytical technique, you will learn about the chromatographic techniques. So chromatography is used for the separation of mixtures from a liquid. So in this way, this is removed, so that we must not have this derivatizing agent in free form in the sample. If it is present in free form, it will create its own spectrum upon absorption of light radiation. We only want this derivatization agent to be present in the form of a complex with our analyte. And now, since this analyte has become fluorescent, we illuminate it with UV light. There will be absorption of light causing excitation. When electron comes back, they again emit light, and the molecule of analyte, which is earlier colorless, will now show light radiation, will now glow because of the derivatized fluorescence method. Now the derivatized sample is excited at a specific wavelength, and its emitted fluorescence is measured at a different wavelength. It automatically comes at a different wavelength. It never comes at the same wavelength. The wavelength of light that is being absorbed, it is always of lower wavelength and higher energy, while the wavelength of light that is being emitted, it is a different wavelength, and it is always a longer wavelength and shorter, and it is always longer wavelength and lower energy. So these are the three compounds that are used as to derivatize a compound into fluorescent compound, to derivatize a non-fluorescent compound into fluorescent compound: anthracene carboxylic acid, nitrobenzofuran, and orthothaldehyde. Now these derivatization methods are used for the following applications or the following type of molecules. As you know that derivatization methods are used for compounds that do not fluoresce naturally. So it is used for the analysis of those pharmaceutical agents. It is specifically used in pharmaceutical sciences as well as medical sciences, because in pharmaceutical sciences, those drugs which do not fluoresce, or those drugs which lacks intrinsic fluorescence, intrinsic fluorescence means a natural fluorescence. If a compound is having intrinsic fluorescence properties, means it is naturally fluorescent. And if a compound does not have intrinsic fluorescence, means it is not naturally fluorescent. And if we are making it fluorescent by adding some agents or some dyes, then it is called as derivatization or extrinsic fluorescence, because we are adding something from outside to make it fluorescent. So those drugs which lacks intrinsic fluorescence, means they are not naturally fluorescent, those drugs can be analyzed by converting them to derivatized compounds which are fluorescent as well, or I should say by adding fluorescent tags to those drugs, and then these drugs become fluorescent, and their concentration can be easily determined in medicines, tablets, or even plasma or blood. Plasma is also the liquid part of the blood. Sometimes it is called as serum. Sometimes we can also call it as serum. Serum is actually the plasma which does not contain clotting agents. If we remove fibrinogen or fibrin protein from the blood, fibrinogen is the protein that produces fibrin which clots the blood. So if that clotting agents are removed, the plasma left is called as serum. So in serum, plasma, as well as in tablets, we can identify those drugs. Even if they are not showing fluorescence naturally, we make them show fluorescence. Again, as I said, they are also used in medical sciences. So in the biochemical and clinical analysis, as we all know that many of the diseases are being tested or diagnosed by various chemical or biochemical methods, or even some clinical procedures. Now, in these chemical or biochemical methods, we basically target some biomarkers, because each and every disease has a biomarker. If the person is having that disease, or if a person is having a disease, or if he is having a disorder, then the body releases some biomarkers in response to that disease or disorder. Those biomarkers, if they are fluorescent, can be detected. If they are not fluorescent, they can be converted into fluorescent derivatives, and in this way they are detected, and if their presence is found, it means that there is some disorder, because their presence is actually a confirmation that the person may have that particular disease. So for that, we can even determine trace level of biomolecules, means the molecules which are present in the body, glucose, even molecules like even some hormones, even some neurotransmitters can be detected. Molecules like amino acids, peptides, proteins, even in complex biological matrix inside the body, they can be detected by adding some fluorescent dyes to those protein, which will go and add to those proteins, and these fluorescent tags are attached to those protein, or they go and attach to a specific site of that protein or the peptide, so that that particular point can be studied with the help of this fluorescent method. Again, even in the environmental samples, if we are, for example, analyzing the quality of water, or even we are identifying some toxic elements in soil, or even poisoning in the blood, we can use this fluorescence method. And if the analyte is not fluorescent, we can use that analyte and convert it into a specific derivatized fluorescent compound by just extracting it, putting some fluorescent dye or some other fluorescent compound like an anthracene carboxylic acid, which then go and attach to those contaminants, and then those contaminants can be detected very easily with spectropluorometers. Again, in food and beverage industries, for example, in soft drinks, in juices, we can identify the presence of pesticides, because some of them are fluorescent. Even if they are not fluorescent, we can make them fluorescent by derivatization methods, or even some heavy metals can be detected by this derivatization method. Then comes the quenching methods, the third method for the analysis of then comes the quenching methods, the third methods, then come the quenching methods, the third method for making fluorescence measurements. These measurements based on the quenching properties, and as we know that quenching refers to the decrease in the intensity of fluorescence. For example, we have a sample that is undergoing fluorescence, which means that electrons are getting excited. When the electrons returns back, it emits light radiation. So for example, if we have this particular compound as an analyte, and if we add a quencher into it, so if the intensity of the sample goes down, means if quenching is taking place, then this means that that particular compound is present. For example, we are adding oxygen gas, because these methods are very useful to distinguish between living and dead cells. For example, if we have one side a dead cell, on the other side a living cell, now these, since it is a living cell, it undergo respiration. In this case there will be no respiration, it will not take there will be no oxygen uptake, while in this case there will be oxygen uptake. So if we add some fluorescent tags, for example this is a fluorescent tag attached to it, while in this case also there is a fluorescent tag. Now if both are giving same intensity, this means that both are showing the phenomena of fluorescence because of the presence of fluorescent tags. But since one of the cell is alive, it is taking cellular oxygen. Because of the transfer of oxygen via the cell, that oxygen will come here and it attaches itself to the fluorescent tag, thereby causing quenching. And when it causes quenching, the intensity of this particular cell goes down. But since in case of dead cell there is no oxygen uptake, and when there is no oxygen uptake, there will be no reaction, there will be no quenching, and the intensity of this dead cell remains same. So these quenching methods can be used to detect between living and dead cells by measuring the quenching properties, by measuring the reduction in fluorescence intensity, and the cell which is showing the reduction in fluorescence intensity, it means it is taking cellular oxygen, and oxygen is a very good quencher. So these methods can be used in that way. On the other hand, if we have a sample which can act as a quencher, for example we have oxygen, we want to detect that whether the level of whether the given water has oxygen or not, and if oxygen is present, what is its concentration. What we can do is we can put a fluorescent compound here, fluorescent compound. That fluorescent compound, when it is used in pure form, it is showing that fluorescent compound, when it is used in pure form, it is emitting light radiation. But as soon as this fluorescent compound is added in this water sample, because oxygen is present here, it immediately quenches the sample, reducing the intensity of fluorescent radiation, which is a confirmation that of course this water contains oxygen, and depending upon the reduction in its intensity, we can identify the concentration of oxygen, and based on that level of dissolved oxygen we can monitor the quality of water, and we can also suggest based on our results, based on the data of this fluorescence spectroscopy, that whether this water is useful for aquatic life or not. On the other hand, if upon putting this fluorescent sample into the water there is no change, or on the other hand, if upon putting this fluorescent sample or fluorescent compound in this water sample there is no change in its intensity, then it is a clear confirmation that oxygen is completely not present in this particular water sample, which means that the level of oxygen is almost zero, it does not contain any oxygen gas, and this water quality and the quality of the water is very bad, and it is not fit for aquatic life. And so further, these quenching methods are divided into two categories: static quenching, dynamic quenching. Static quenching refers to the quenching the intensity of light even before the emission happens. It means these static quenchers go and attach to the analyte even before excitation occurs. So there will be no excitation, there will be no deactivation, and when there is no deactivation in any form, neither in the form of light, which is called as fluorescence, nor in the form of non-radiative emission. Static quenching refers to the formation of a non-emissive complex, means the static quencher go and immediately reacts with the analyte, and it does not allow the analyte to emit light radiation, means it goes and react to the analyte or to the specific fluorophore, to keep it in ground state, preventing the emission before excitation, preventing the excitation, even the excitation does not take place. While in case of dynamic quenching, the quencher reacts or restricts the light which is being emitted. Means in case of dynamic quenching, there has been a reaction. There means in case of dynamic quenching, the analyte or the fluorophore absorb light radiation, causes excitation of these electrons from ground level to excited level. When the electrons reaches to excited state and our analyte becomes an excited analyte represented by A star, at this point only the quencher comes and forms a complex, so that there will be no radiative deactivation, means no light is emitted. But in this particular case, there may be some energy loss which may be due to non-radiative losses. So this is the main difference between dynamic quenching and static quenching. Static quenching refers to the process in which the quencher goes and make complex with the analyte in the ground state only, so that it will not create an emission spectrum, so that there will be no emission. While on the other side, dynamic quenching refers to the method in which the quencher goes and react with the analyte in the excited state only, because at that particular time analyte is already in the excited state, and when the quencher forms a complex, there will be no emission of light, there will be no radiative deactivation, but a non-radiative deactivation occurs in this particular case. And therefore dynamic quenching is very useful to study the folding and unfolding of protein molecules. When there is an unfolding in the protein, there will be light emission. When there will be when there will be unfolding, there will be light emission. When there is folding in the protein, there will be no light emission due to dynamic quenching. So these dynamic and static quenchers are used to develop sensors, fluorescence sensors. So these dynamic or static quenchers can be used to study the fluorescent sensors that are useful for detection of various compounds. Quenching methods are applied for determining the binding affinities of proteins. For example, if we have a protein or a phenolic compound or any other compound, and we have added a quencher into it, and if that quencher goes and attaches itself, we can identify those binding sites and the binding constant, and this is confirmed by the reduction in the intensity. For example, if the protein is showing some sort of fluorescent, we add a quencher over there, the quencher goes and react to that site, and ultimately there is a reduction in the intensity, confirming the binding between the quencher as well as the protein or any other compounds. These quenching methods are also used to quantify the concentration of a substance that act as a fluorescence quencher. For example, we have a sample in which some quenchers are present. For example, in case maybe oxygen is present as a quencher. Now when we add a fluorescent compound into it and the intensity of fluorescent emission decreases, this shows that there must be some quenching agent present in the sample, and if there has been no change in the fluorescent intensity, it is a signal that there is no quencher present in the sample. Again, molecular interactions can be studied by these quenching methods. Especially the dynamic quenching methods are useful for studying the folding and unfolding of protein molecules, because proteins are basically polymers of amino acids. There is unfolding of protein and then there is folding of proteins. Large molecules occupied a very small space by folding. So this folding and unfolding basically confirms this folding and unfolding can be confirmed by the intensity of the fluorescence radiation. For example, if upon adding the quencher there has been no change in the intensity, this means that in the present state the protein is in the folded form, and when protein is in the folded form, that site is not visible for the quencher to go and attach to that site, causing quenching. On the other hand, if upon addition of that quenching agent there has been a decrease in the intensity of light and quenching is taking place, this shows that the protein is in unfolded form and those sites are clearly visible, and those sites are clearly visible and approachable, so that the quencher goes and very easily attach itself to these sites resulting into the decrease in the intensity of the fluorescence radiation. And last, we can also study the biomolecular structure as well as dynamics. This means we can study the local environment of a fluorophore. Depending upon the local environment of a fluorophore, that fluorophore responds upon irradiating with UV light. For example, if a fluorophore is there and we are illuminating it with UV light and the environment is fine, there is no quenching agent or some other compound present, there will be normal emission of light radiation in the form of visible light, which is called as fluorescence. But if there are certain compounds, if there are certain groups that can act as a quencher, or even oxygen or some other compounds or even some metal ions are present near to that fluorophore, then the presence of these oxygen molecules or even metal ions, they go and attach to the fluorophore, resulting into the reduced intensity of this fluorophore. So this method can also be used to study the local environment of the fluorophore. Again, DNA hybridization can be studied. This is through fluorescence quenching, which is a key component in some methods to detect DNA hybridization, where a fluorophore and a quencher are placed at opposite end of the single stranded DNA molecules that come together upon hybridization. Means we have a DNA molecule like this, and we put a fluorophore here, which is a fluorescent compound, while on the other side we put a, so this, we have a DNA molecule like this, and this is a double strand DNA molecule. Then we have a single strand molecule here. So we put on one side a fluorophore, on the other end we, at the other end we put a quencher. Here we put a quencher, here we put a fluorophore. And if the intensity of fluorescence light remains same, this means that quencher and fluorescent molecules, they are far away from each other, which means there is no hybridization, there is no folding. But if the intensity goes down and quenching is observed, this means that they are getting close to each other, and in this way quencher forms a complex with the fluorophore, thereby reducing the intensity of the fluorescent light, which is a clear indication that folding is taking place or hybridization is taking place. So these quenching methods are very useful to study the biological systems, and they are also useful in environmental samples as well as in medicine. So with this, I am going to end this lecture. Thank you very much, and in the next lecture we'll discuss about the applications of these fluorescence methods, and we'll also solve few problems relating to fluorescence spectroscopy. Till then, take care. Thank you very much.